Now, after understanding the concept of the microscope, let's look within the microscope and understand what are the different structures that are possible to be visible. Now, one of the most common one that you can probably see within a microscope is the membrane. A membrane is mainly a boundary outside the cell. So you will be able to recognize membranes or cell structures or a boundary when you look into a microscope. Now, each membrane is composed of the macromolecule. If you remember earlier when we discussed the lipids, I said the lipids were had a head and a tail, that is a non-polar tail and the polar head, and they are organized in this scenario, which is shown over here in these images that you see over here. Now, the lipid would be, as you know, is not water soluble, so it would prevent certain substances to move through the membrane. And this is solved by the presence of certain proteins and carbohydrates that are embedded within this layer. The layer of the membrane is a fluid mosaic. A fluid layer is something, for example, when you have if you take water in a pan and you drop some soap in it and you see the soap moving or floating in water, that's a consistency of the membrane. So it's a very fluid, dynamic, living membrane. And the membrane is composed of also, besides the carbohydrates and proteins, it sometimes also has steroids and phospholipids embedded in there. And these pretty much help in the different properties of the membrane that we will see later. So within the membranes, I mentioned that there were proteins. These membrane proteins have a very vast role in the cell's life. Some of these can be receptors. Receptors could be certain region of the membrane that will respond to a hormone or a signal coming from another part of an organism. Some of these proteins can be transporters, which means that they will help take something that was made within the cell and transport it to a different part of an organism. Some of these proteins can be for communication and signals. And lastly, these proteins can also be for adhesions. For example, in some sort of connective tissue or histology slides, when you see numerous cells that are of the same kind and they are arranged in a row, they have to be glued together, and these are the adhesive properties of these proteins. Lastly, but not last, the most important is it holds what comes in and out of the cell, and these are the recognition proteins. Just like if you have if you live in a home and there is no doors, you really have no way of interacting with the environment. So these doors that are present on the membranes are mainly protein, and they guard what comes in and out of the cell. So they are very recognizable proteins. Now, when earlier in the beginning of this topic, we talked about prokaryotic and eukaryotic. Now we will explore prokaryotic cell a little bit in more depth. Prokaryotic cells are very early cells. These cells are present in the kingdom or the domain archaea, and also in the domain bacteria. Now, within the bacteria and the archaeas, the kingdom level has not been established yet, and they're still working on it. But these are the simplest cells. The simplest cells are mainly made up of a cell membrane, which is the boundary, and different regions that carry out various roles of the cell. For example, creating energy, making manufacturing certain food, recycling, even cell division, passing out certain messages and toxins. So all of these activities are carried out within this enclosed simple primitive cell, and there's no compartmentalization in there. So if you think of a big room or a studio apartment where you have everything together and no organization, that's your prokaryotic cell. In a eukaryotic cell, there is more compartmentalization. So you have a region which is your, let's say, your bedroom. There's a region which is your TV room. Then you have a library, or you may have a den room. So depending on what kind of a eukaryotic cell, it can go very extensive. Prokaryotic cells are also one of the places that you find them are example on your teeth. There are lots of living organisms that are arranged, and they constitute what is called as a biofilm. Not all prokaryotic cells are harmful to us. Lots of prokaryotic cells have very important functions within human bodies or other multicellular organisms. The prokaryotic cell has another region outside the membrane, and that is called as a cell wall. A cell wall will surround the membrane and give it more structure and more support to the membrane. Certain bacteria will have cell walls in which protect the bacteria from being destroyed by certain chemicals. So it's more like a survival mechanism for a bacteria. Certain bacteria may have hair-like structure which helps it to move or to locomote, and these are called as flagella. So these are going to be structures that will help for the motion of these particles, because mainly these particles are living in a fluid environment. Besides that, there are also certain, I'm going to make another bacterial cell over here and show you what a pilus or a pili are. These are like connections between two cells, and the genetic information can transfer from one region of the cell to the other. I don't know if you can see very well on this image over here, but these are all hair-like structures, and they're all flagella. This is an image of the biofilm. Again, cell wall forming a very resistant structure in K. pneumo. And here are certain prokaryotic cells that are shown that are arranged in a bead-like manner. So what are eukaryotic cells? Eukaryotic cells are those cells that have very rigid compartmentalization. This image on the bottom is a plant cell, and the one on the top is an animal cell. How was I able to quickly recognize that, besides taking biology for years? Is because of this space that you see over here. This space is a vacuole which is very predominant in a plant cell. It almost sometimes takes 70 to 80% of the volume of the cell. Now, with that, besides these, you can see these structures that look like regions that are fixed for certain function, and these regions are because of what are called as organelles. Organelles are membrane-bound structures, and they're very specialized in carrying certain duties within the cell. Nucleus is one organelle, endoplasmic reticulum, Golgi bodies, vesicles, mitochondria, and chloroplast. There are certain organelles that do not have membranes, and those are ribosomes. And besides that, there are other structures that are present in eukaryotic cells, and those are cytoskeleton. So the first coming couple of slides are going to review these first structures that are organelles with membranes. So let's look at the first organelle that has membrane, that is a nucleus. If this was my large cell, then you can see the nucleus is this region which is enlarged in the second image over here. In this image, you can also see something very clearly, and those are the black spots that are present. And these are the nuclear pores. These are little holes that are present on the membranes, which helps genetic material to come in and out of the cell, so the direction from the nucleus can be given out to the cell. Nucleus is your command center of the cell. It holds all the genetic information. It controls all the activities of the cell and how the cell reacts to the environment. Within all this information is usually present in what we call as the DNA. DNA is theoretically is a very long, fine a polymer of nucleotides, and these polymers are then arranged around regions that are called as proteins, which help it to fold, and eventually they get folded and they look like thick structures, and these thick structures are called as chromosomes. So chromatin is your long elongated fiber-like structure. Your condensed form is chromosome, and this condensed structure is carried around the proteins, which help it to give a main stem that the fibers can be folded around. Within the nucleus is also a region which is called as the nucleolus, and the nucleolus is a region that produces or manufactures ribosomes, which are other structures very important for manufacturing proteins, but the ribosomes are synthesized within the nucleus. The nuclear membrane is a two-layer membrane, and you can see over here this is an opening which is called as this is the nuclear pore. Now this membrane is very interesting. The membrane actually starts invaginating, and it starts making structures outside the nucleus. So let me show you on the next slide. So if this was my cell and I had a nucleus over here, the nucleus is two membranes, and the second membrane starts coming out in these folds, and it forms what is called as the endomembrane system. And this endomembrane system is very extensive in eukaryotic cells. And on the endomembrane system, there are sometimes beads that are present, and these beads are the ribosomes. Remember previously I said ribosomes are a place where proteins are going to be synthesized. And these endoplasmic reticulums that have these ribosomes on them are called as the rough endoplasmic reticulum. The R stands for rough, not for ribosomes. The other ribosomes that do not have the rough endoplasmic ribosomes studded on there are called as the smooth endoplasmic reticulums. And the smooth endoplasmic reticulums forms lipids within the cell. Now besides that, there's another endoplasmic reticulum, and those are the Golgi bodies. The Golgi body is mainly a packaging center for the cell. So it's a center where the proteins or lipids that are formed are first stored, they're packaged, and then they are transported. Sort of similar to what UPS or FedEx would do. So that would be your Golgi bodies. Besides that, what else is there and what else goes on within these membranes are, these membranes sometimes help to transport proteins and lipids in and out through a process of endo and exocytosis. Endo is in, so you can see in this image how the membrane is invaginating, and eventually it will close, and this structure could then be exported out. And if you can see over here, these round structures are showing that how the proteins or lipids have been exported out, and when they can come back and they adhere to the surface, how these membranes can attach to a new surface, let's say this way, and how it will open up that surface and then slowly start dissolving these membrane proteins and eventually pushing the substance in. And there's a nice animation that you may want to watch for this process of endo and exocytosis. Besides, there are other vesicles. One of them is called as the peroxisomes, and the second one is the lysosomes. Both of these are like your recycling centers. They're like your trash removal. With all this chemical reaction that are going on within cell, there's a lot of toxins and metabolic products that are waste, and these have to be either recycled or reused or totally removed from the cell. And peroxisomes and lysosomes would do that. And remember, the cell walls of the plant cells indicated that there was a central vacuole in the plant cells. And the central vacuole, I'm going to show you back on a slide over here that we saw previously. This vacuole is filled with water or fluid, and it gives a very strong turgor pressure to the walls of the plant cell.